Please join me in welcoming to the Distinctive Voices podium, Dr. Carla Pugh.
Well, good evening. Nice to meet all of you. Thanks for coming here this evening and allowing me to share some of my work with you. So my goal this evening is to tell you a little bit about my inspiration for the work that I've done, little story about the first simulator that I developed, and then get into the sensor data that has come out of this work, and talk about some of the surgical simulators that I've developed as well. When I think about what really had me take a detour from my surgical training, once I finished my residency, I went to get a PhD in education. I was very frustrated, to be honest, with my surgical training. And this picture represents part of the problem that I experienced, and that it became clear to me that I could not learn everything that I wanted to learn simply by watching someone do a physical exam or do an operation. There was a lot that I could learn, but I knew I couldn't get to mastery, and that's what I wanted. This is a more detailed example of one of the experiences I had as a second-year resident. I was rotating on the urology service, and I was going to do or assist in an operation the next day on a patient that had an enlarged prostate. And this is another picture to show. It was the surgical procedure was called the suprapubic prostatectomy. And although they weren't really removing the prostate, they were removing a small tumor, a growth in the prostate in this area through the bladder. And I was very excited because I'd never seen one before. And I have to say, I had not seen this picture. There was no YouTube or Google image database that I could find these pictures in. None of the textbooks had a picture of the anatomy and what it looked like. So I just had the netter anatomy of the bladder, and I found a few pictures, but I really didn't understand exactly how this was going to work. So I stayed up all night reading and looking through a bunch of textbooks and surgical atlases, and then I got to the operating room, and that's what I saw. For 2 hours. Obviously, I was very disappointed to be looking at the back of the surgeon's hand for 2 hours. Probably an hour into it, and I'm a second year, so I'm not supposed to talk. I train on the East Coast. It's very militaristic, so you're not supposed to ask questions. So I knew better to talk to the attending, but I looked at the fellow and the resident who was there, and I finally just broke my silence. I said, "How are you learning what he's doing?" And the room went silent. I'm like, great, I'm getting in trouble now. This is, And the surgeon, the resident didn't say anything. He just looked at me because I spoke. And the surgeon then took my hand. He said, "Here, I'll show you." And he shoved my hand in the pelvis, and he says, "You feel that little ball? It's attached by a little string there." He said, "That string is a vessel still attached to the tumor I'm trying to get out, and that's why I'm really being careful here." And I'm like, okay. Thanks. He's like, "Thanks for asking. No one's ever asked." I'm like, "That's because you always don't allow us to answer questions." But that was actually an everyday experience at some point, whether I was watching a surgeon do a breast exam or do other procedures. Some of the medical exams and procedures, you could see the person's hand. Some of them you couldn't. But it was still clear to me that I wanted more information if I was really going to learn what they were doing. So this is what led me to the farm, to Stanford after my residency to get a PhD in education. And that's where I learned about sensor technology. And that opened my eyes to a lot of possibilities. The first simulator that I developed was I took courses, even though I was in the School of Education. They allowed me to take classes in computer science and other areas of my interest, and I took a class on human-computer interactions. And this was a class where it was a competition, so our first laboratory experience was to propose a project and then get someone, everybody had to propose a project, but you had to give such a good presentation that someone would drop their project and join you to develop yours. And so this was a competition, and of course this was not my only class, so we were all very busy, and I remember finding myself in some super mega store at 2 a.m. trying to figure out what I could buy to build what I wanted. So I'm roaming the the aisles in the store, and this particular store had a sporting goods section. And the first thing I saw was a badminton birdie. And I'm like, okay, what can I do with that? And then I got an idea immediately. So then I got a toilet paper roll where I had to buy full toilet paper and then take it all off and get the end roll. And silly putty and saran wrap. And what this was a cervix simulator. So I had to give the engineers an understanding that you've got an internal organ that you can't see what someone's doing when they're trying to touch it, and it's at a distance. And then I put a sensor on the end of the badminton birdie, which served as the cervix. And that was my first simulator, and given that it was this sort of generic presentation, I got some brave soul to join me in my project. And later, an OBGYN professor gave me a mannequin model that I was able to instrument with sensors, and then that ended up being my patent that I got relating to the use of sensors. I didn't patent the pelvic simulator, I patented the use of sensors for hands-on examination to capture and display that data. So you can tell this a long time ago when you look at that laptop. But this is the first simulator I built. And basically what we did, and this is not one of the sensors I used, this is another sensor. The ones I had were a lot smaller and really paper thin, and we mounted them on various organs. These are 3 organs, internal organs that come with the model. So the model looks the same on the outside, but the skin comes off, and you can change the internal organs. And this is, if I can get, well, the one at the top is a normal cervix and uterus. The cervix you can change, and that had some abnormalities or options for abnormality. Obviously, the one below is a horribly abnormal uterus with fibroid changes. And what I did was put sensors on the model, and this was the resulting interface that my colleague helped me to develop. And basically, you can see where someone is touching and with how much pressure. All of the sensors can be activated at any given time and captures the whole examination from beginning to end. And this is an example of some of the data. It's like less than 1 second worth of data, but the highlighted area shows a sensor going up to 6 pressure units, and it goes back down to normal. But this is the data that I was able to collect from the model. And that's pretty much been the source of my research, really raising the question, what does this mean in terms of performance? And so I've collected data on several thousand people over the past 14 years looking at this, and I've also instrumented other models. But this is a line graph representation of the data, and each of the different colors represents a different area of the anatomy. So the pink area is the cervical os, which when you're obviously any OBGYN docs in the audience, that's what you're supposed to feel when you're trying to figure out how dilated the mom is and if it's ready for the baby to come. But that's, you can see how many times and how they've touched the cervical os during this procedure. But for my dissertation, what I looked at was the time that it took to do the exam, the number of critical areas, or really number of sensors touched during the exam, the mean and maximum pressure for each area that was touched, and the frequency. And that gave me some idea of how to characterize what someone was doing when they're doing the exam. One of the things is we were trying to really get at another level to understand what was going on. We started to notice, I had a data mining professor at Stanford say, well, why don't you do an experiment where you do movements that you normally do, and just focus on that movement and see if we can see some patterns. And that was actually eye-opening for me, because this is what it looks like if you're pressing in one place up and down, and this is what it looks like if you're pressing up and down in a small circumference. And you can see the pressure goes down to 0 when you lift your finger all the way away from the tissues that you're pressing. Circular and rubbing motions look a little different. And that's what it looks like if you're rubbing doing a movement in multiple small areas within a radius. And so that actually really helped me to go back and look at the line graphs and understand what people were doing, what types of palpation techniques they were using. And this was very exciting for me. And once I learned how to package the interface and build other ones, and also the sensors, I sort of instrumented every mannequin I could get my hand on. And so I'll show you some of the data. One of the ways to look and see if this data or information will have any use is to look at known groups, and see if there's any difference. And so I looked at medical students, residents, and experienced clinicians performing a rectal exam on one of the models. This is the model actually being used in a classroom. When I was a professor at Northwestern, I used it in the classroom for teaching, and you could see how the students are doing an exam. All the models look the same, but there's different organs on the inside. And they're all looking to the side at the computer to see where they're touching and with how much pressure. For the experiment that I did, if this is sort of the normal, it's hard to explain, but if this is what a normal rectum and prostate looks like through an anoscope, the first model that I presented was one that had a fairly large, hard, abnormal lesion on the right lobe of the prostate. The other one had bilateral enlargement, which is supposed to represent benign prostatic hypertrophy. And then they also had a soft rectal mass, exactly opposite the prostate. And so I presented these two as an experiment to a group of students, residents, and practicing clinicians. And I'll give you the results for the more difficult one. The easy one, everyone got it right, and there was no major or significant differences. But for the very difficult one, these are the results. When you look at the students, they actually spent less time doing the exam. And so there's some interesting things. I noticed changes in the learning curve. When you look at the residents, they spend the most time, faculty in between. Faculty touch more areas than the residents and students with greater pressure and more palpation frequencies, and they're more accurate. The students had a problem with their accuracy, a significant problem with the prostate. And when I got to interview some of the students and sort of understand what was going on, it was clear that they actually had a misconception of how to do the exam. Those students in this experiment that went from learning it in a textbook to being tested, and some of them had already done exams on patients, what they saw when they look, not using the simulator, because I hadn't given them results yet, but when I tried to understand how they learn the exam, mainly by watching videos and watching professors do the exam, and what they see is the back of the clinician's hand doing this. And so that's what they thought they were doing. This is the problem. The students were doing this, and just with a straight finger, whereas the clinicians were doing this. And so the students were missing lesions on the sidewall because they weren't palpating and doing a full exam. They just only saw this. It was another reason why their exam was shorter, because they weren't really doing anything. So we let the news out. We published a paper and presented it nationally and exposed that some of the students really don't understand this exam, and that helped us change when we incorporated the simulators into the curriculum. We let the instructors know, make sure the students understand the proper palpation maneuvers. And this was just the data showing the layout of the number of students that actually touched the lateral aspects of the prostate. It wasn't many. So the next thing we did, I said, well, this is actually pretty good. I can tell the difference between known groups, novices, and I can also understand where they're missing it. So then we took it to a larger meeting, actually 2 large meetings, one to get data from colorectal surgeons who do the exam regularly, also urologists. These are a group of urologists, and it's actually interesting to set up an exhibit hall. Hundreds of doctors line up because they said, my exam is the best, you have to have me in your database. And I'm like, okay, great, come on. And then we also did it on a final exam at a medical school. So we got a fair amount of data, used the same models that I had before. And this is the data on the easy model. No significant difference in terms of their accuracy, all above the 90th percentile, but not the same for the difficult model. The urologists were pretty on point with the benign prostatic hypertrophy. The colorectal docs and students, not so much. This was pretty interesting. So I found out that probably about 50% of the urologists don't do the rectal exam when they're examining the prostate. And that's a huge problem because you sort of just assume that they do a full exam while you're there. But some of them never, they go in, they examine the prostate, and they never supinate their finger. So some of them, and I could tell because those who missed it, the sensors didn't register on the rectal mass. So that was news to us. I think that represents a whole nother problem for us because when you have primary care physicians, you know, the rectal exam is not an exam that you want to have 4 times a year. If you're going to get one and everything's normal, you prefer to have the best and most complete exam that one time. When I think a potential problem here is that if an internal medicine physician sees a patient and they have urologic symptoms, then they say, I'll spare you the exam because the urologist is going to do it. And then they go to the urologist, but the urologist only does a partial exam. And I've had patients come to me after 6 months of getting other procedures, other workups, and then only to find out that someone didn't really do a good rectal exam and they've got a rectal cancer that's actually causing some of their problems. So I think the excitement for me with finding this is that I think this technology can also sort of help us understand natural changes in practice patterns. And I think what's happening is the urologists are just getting super, super specialized. Everybody's picking their own organ, but a lot of the generalists think that everybody who does the exam is doing a good job, and that's not the case. On the other end of the spectrum, this is this is some data showing, you know, the sensor works as well with the airway. And this is a sensor output from an experienced anesthesiologist doing an intubation. And I don't know why my pointer isn't working, but the red or brown line at the top is actually the sensor on the tongue, which is where the laryngoscope rests. And when you see the blue line, that's actually when the intubation or the breathing tube is being passed through the vocal cords. So you can see that this person took one swipe, and then the sensor reading went away, and that's because they put the instrument in, and then they turn to the side to get the intubation tube, and then they hold constant pressure and then slide the tube in. This is a novice exam. And the first thing you notice, I actually had to squeeze it, thank you. I had to squeeze the graph to fit it on the screen because their intubation took 3 times as long. And when you look here, they did several manipulations of the laryngoscope, and you can see the pressure going up and down. Then they held pressure, and then you start to see sensors that you never saw on the experienced intubation. That's because they're doing extreme tilting of the neck, and so actually areas that you shouldn't be touching or you're seeing pressures, you start to see that here while they're holding pressure and trying to get a good look but wasn't able to pass the tube. They manipulated the laryngoscope again several times and then finally got the tube in, but they really didn't have good control of the airway, and I'm sure that you can get injury to the hypopharynx and the teeth and other places. And so now I have if you look at how we would traditionally have graded this resident, we would say, okay, great, you took a little longer to get it in, but you got it in. That's not good enough, right? We need a little more detail. So now I have more information and I can show specifically and objectively, you were doing a lot more tilting than you needed to. More than likely they were holding the laryngoscope in the wrong way such that they weren't able to visualize the airway as well. But this is very helpful in terms of giving detailed feedback. What's fun for me is that by this time, when I had instrumented at least 3 or 4 models, I had enough preliminary data to submit a grant to the NIH, and I got funding to do a huge project with the breast exam, and I'll give some data from that. What was also really exciting was the chance to have some consultants at the national level who are experts in metrics, and they do this for a living in terms of determining how to use data to assess performance and to establish minimum competency. So this is a fun group of people to work with who are very knowledgeable in this area. And this is, what we know is that there are several different ways to do the breast exam. Initially, I didn't start with the breast because I know it's more difficult. If you think about it with the pelvic exam and the prostate, you only have one finger or two fingers and you've got limited degrees of freedom. With the breast, you can do anything. And so I knew the data would be more complex, and I started with the basics, just looking at, if you see this diagram, the colors are where we have the sensors in the breast model, and this person is doing what's called the lateral strip method. And that's what the exam looks like on the sensor data using the lateral strip method. This is what it looks like doing the circular method. And so just in looking at that, I can tell the difference in terms of what approach someone is taking, and that gave me some confidence that I may be able to get some interesting results from the breast exam. I also, with the R01 grant, upgraded all of the technology that I was using. So instead of using individual sensors, I think when I was a grad student and first instrumented the breast model, I had 11 sensors. I then was able to get a distributed sensor, a sensor plate that has over 2,000 sensor elements, so I could get more detail. I also then started collecting video data so that I worked with a professor who does post-processing of video for motion tracking. And so this is our setup our new setup for the breast exam as of last year. And we've collected a fair amount of data using this setup. And the way they do the motion tracking is that they can actually select a certain pixelated area, and then they can follow those pixels to get an understanding of motion. Then the sensor maps, this is how the data shows, and this person is actually doing a lighter palpation at this point in time. I think I'm breaking the, it's me, it has to be me. That's so funny. It's okay. So the darker red means there's more, the deeper the palpation. And this video, which I hope will play, what was funny is, again, I'm trying to get data on things that we've done in medicine for hundreds of years, but we never had a language for it. So I had to make it up because I have to code my variables once I put them in an Excel spreadsheet or for SPSS to analyze the data. And we noticed some technique where people they sort of do different maneuvers, and I'll just show you because all of them will play at once. But this person in the person number 8, does more of a circular movement, and he's really quick when he does it. Person number 107, upside down, doesn't do any rubbing at all. She just sort of moves forward and backwards. The other linear rubbing, number 28, he moves in this direction. So I'll play it a few times so that you can watch. And the other person is interesting, does something different. So. And after looking at 100 breast exams, probably almost everyone does it differently. And so that then made, obviously, my research a little more complex in terms of trying to figure out what's a good exam. Again, the finger movement person we called the piano man. They're just really going, the other person doesn't move their fingers at all, doesn't do any rubbing, but is doing the linear strip method. The circular rubbing guy not only moves circular, but he's also going in a circular pattern. So there's 2 levels of information. It's, the pattern that they're using, but then also the local finger movements that they're using. And we're, with this new sensor, we're actually able to get some information about that. This is what we're finding. One of the things that we've gone to 2 meetings so far. We went to a breast surgery meeting, and got data from 140 surgeons, and then with 2 different models, breast models. One that has both of them had an obvious lesion. One was superficial but very soft. One was deep, 2-centimeter lesion, but it was rock hard lesion on the chest wall. And when we started to look at the differences in how people do the exam, we started to know there's an association between the amount of pressure you apply and accuracy. And we didn't have a term for some folks, but we could notice on the video as well as on their pressure map, we call them the patters. And they're never going to find anything. And it's very clear, and they just do the exam with very soft pats, and the other people some people start patting, but then they go deeper. The ones who miss it, they stay patting. And on one of the masses, 90% of the people found it. There was only 16 in one group that didn't find it, and we looked at match controls to see what the difference was. And it's pretty clear that pressure is a huge issue. If you look at below 5 newtons, none of the people who found it had pressures that light, and even only a few below 10 newtons. But it's a big difference in terms of the amount of pressure that people are applying, and this seems to be an independent variable associated with accuracy. Obviously, I'm interested in those 5 people that were below 10 newtons, that they used, a moderately light pressure, lighter than the rest of their colleagues, but they still were able to find it, which goes to perception. So there's some other elements to the exam that we're learning. In all of this work, I have come to believe that there are some people who have a haptic, and this is this sense of touch. Some people are actually really gifted, and other people I think are probably haptically challenged and probably shouldn't do breast exams or have professions that require you to have a high level of sensation at your fingertips. So it's been interesting watching novices come in, and some of them have never done the exam, but right away they're like, wow, this side feels really weird and that side doesn't. And others have no concept of the difference between two things that we're showing them. So it's been pretty interesting. So I'll move on to the surgical simulators, which is my sort of second part of my story here. So I obviously am a surgeon, so I've been interested in developing simulators for surgeons, and I worked with a cognitive psychologist who came and interviewed several of my colleagues for different procedures that I wanted to simulate. And I also spent a fair amount of time with a bunch of engineers and companies that design and develop simulators. And it was clear that if I wanted to assess or train high-end surgeons, that what I really needed, I didn't need a simulator that they could, unless it was the Star Trek holodeck, that they could do an operation from beginning to end. What I really needed was to situate them at a critical decision-making point in an operation. And so when he did these interviews, he did a cognitive task analysis approach and was able to pull out what were the critical parts of the operations that they were a specialist in. And then I built a simulator focusing just on that part of the operation. This was one of the more basic ones where you're taking a piece of the intestines and bringing it out to the abdominal wall. We do that procedure for persons who have had an abdominal emergency or that have a really low-lying cancer, colon cancer, and they're not able to have their intestines put back together. You have to form a colostomy or an ileostomy. And this is, these are pictures from a video. This is one of my colleagues at Northwestern, and I am interviewing him about his approach to doing an ostomy. But these are fabric-based models that we've built and then farmed out to a company that now makes them. And he goes through the process through the layers of the abdominal wall and then matures the ostomy. And that's where the layers are. There's a difference in terms of how you mature a colostomy versus an ileostomy in terms of the thickness of the bowel. This is another simulator, and I wanted to get one procedure that was within a range of surgical techniques that we use. So this one's a median sternoscopy, and what's fun about this is the surgeon was able to bring his instruments from the operating room and perform a lymph node biopsy. He's looking upwards because this is a video procedure, and he's looking at the screen, and then he looks in directly once he finds a lymph node. And this is an inside view from his scope, and we made lymph nodes out of bunch of different materials, seeds and fake blood and an airway, and the blue thing there is the innominate artery. So and it's filled with blood. So if he actually accidentally hits it, then he'll get some bleeding and have to deal with that. That's another view once he got a sort of a ring around the lymph node, and then he was able to pull it out. This one is for a different procedure, a laparoscopic procedure for someone that has a hernia of their abdominal wall. This one's a pretty big hernia. I don't know if you can see, unfortunately, in the distance, there's a little lump there. But again, what's fun with the fabric-based simulators is that you're able to use all of the same instruments that you have in the operating room. I have played with some of the VR simulators, and there's pros and cons to using physical models versus virtual reality models. I can actually make the physical models more difficult than the virtual reality models. The graphics are better on VR, and also you're able to collect data. But in this instance, I'm able to do a lot more in terms of clinical variation than is easy enough to do with the VR products. This is a close-up of the hernia and one of the surgeons in a course repairing the hernia, and this is the setup. We set it up like a real operating room with a patient, and they're using the same instruments that we use in the operating room, except for the cautery, which then the patient would catch fire, and that wouldn't be fun. So that's the only instrument we can't use. This one is even another procedure, which is sort of high-end, a pancreatic jejunostomy for pancreatic cancer. And this was a prototype that we were testing. Obviously, this patient only has one segment of intestines, but we wanted to see proof of concept, and this is a pretty well-known pancreatic surgeon who's doing a procedure on this model, and this is a close-up view of all the millions of stitches that they use, and it's a sponge. And it costs 5 cents to do this, and what I learned more about sponges than I ever thought I would. There's a wide variety, and this one was perfect for a pancreatic cancer because it was a little more firm. We had another scenario where it was a very soft gland because the cancer was in the duodenum, and that's more difficult to sew on because once you put your stitch in, when you tie, it can actually tear through, and that actually can happen in the operating room. But this one has a fairly large duct and it's firm, and he's showing sort of the complexity of all the sutures that they have to put in through the back wall and then the anterior wall to the bowel. And it actually gives us an idea to understand how much residents really remember in terms of how to organize the gland and the sutures in terms of lining everything up perfectly. Because if you don't do it right, then it will leak. And the pancreatic juices are pretty caustic and can cause problems. So if I can sum up the research, if you will, that's been done with even motion tracking and sensor data when we are trying to learn how to use simulation technology for testing and teaching, this is pretty much it. That the experts have smooth signals, and this is someone who has motion tracking sensors on both of their hands, and you could see there's 2 signals, one on the left and the right. That's a left and right hand for an expert. What we know consistently, regardless of the procedure, regardless of the technology that you're using, is that the novices have messier signals, and that's pretty consistent in any of the literature that you read. And I think the question is, what does this still mean in terms of performance? Where do we go from there? And what I did again, which is my favorite thing to do, is start with the basics so that I can understand where we go next. And this is one of our experienced surgeon who has some motion tracking LEDs on a surgical glove, and we're having her do a simple suture. But what I did was a little bit of trickery in that I gave her 3 different tissues to operate on. One was a balloon, the other one was soft foam, and the other one was toilet paper. And in this experiment, what I was purposely trying to see the differences in motor planning. And I know that when you look at what affects decision-making, it's perception. And we also see a range of these types of tissues in the operating room, and we're always doing a calculation and an estimate of what's going on. We are, if you have a pancreas gland that's more fibrous, then you're a little more comfortable because you know it's going to hold the sutures. If the gland is soft, then you're moving a little slower and a little more controlled because you don't want to tear the gland, and that can be a problem. And so I wanted to put the surgeons in a situation where they had to adjust to their perception of the tissue and what needed to be done in order to get it right. The surgeon did pretty well. The medical student didn't. And there's a number of problems here, even just the difference. The student, when you're using this type of suture, which is Proline, it has memory, you usually leave at least a centimeter or two of a whisker on the end. So not only did the medical student not get the stitches in, but they cut this suture is just going to unravel at any moment and burst. But, when I look at this, again, what can I tell this student? So when we look at the motion tracking data, there's a lot going on there. Again, you can pick out the expert and the novice, right? The novice has the messy signal. And when you look at these, this is really the displacement. And this person is doing these big, wide movements with the suture, and they really have too much going on. And you can't do that when you have soft tissue. You've got to be very careful and focused with your hands and movement. And so this is why the student didn't do as well, because it didn't have as much motor control. What's also been interesting in looking at this is the reality is that there's a lot of decision-making in surgery, and a lot of people think that it's more just the technical skill and the craft. And also, I could say a lot of my colleagues who are doing some research in this area still focus on the motor aspects in terms of the movements. And what we're finding is that there's more to the data, that the periods of where there's non-motion are actually probably just as important or more important in understanding expertise. And when you look at just one stitch with the novice student in black and an expert in red, you can see not only the difference in the displacement when they're starting, but the difference in pauses. And the expert just has sort of a smooth transition in terms of getting the stitch in. The novice is doing these big movements, then there's a long pause, and then there's posturing and another pause. This whole segment here, a little bit of movement, but that's they're displaced and they're just standing in space, trying to decide what to do next. And we found a way of analyzing when there wasn't motion, and we're starting to see that we can get an indirect understanding of motor planning and decision-making and expertise when we're looking at idle time, initiation velocity when they first start, how quickly do they start, as well as the number of times they start and restart, and reaction time. So I'm pretty excited about looking at this other part of the data, and we're presenting a paper at the end of next month to show that there is a huge difference in these four variables and looking at known groups, students and faculty. So this is sort of a new area in terms of looking at areas where there's not movement as opposed to just the motor movement. Obviously, there's parallels, and this always strikes me, I think, for us as surgeons and people who are in medicine who do procedures, that we are kind of lagging behind, if you will, compared to other industries who use a lot of technologies and videos and things to perfect their skill. And so we've got a lot of work to do. But this is, if you're wanting to get to mastery, it's not only things that can be observed, but it has to be measured. And that's the way that you're able to give real feedback to someone. Again, this is, again, looking at what surgeons do in the operating room is not only what you do with your hands, but also with your instruments and how you handle the tissues when you're doing an operation. And I think when we start to look at how we define a master surgeon or an excellent surgeon, we're still in the mode of using subjective peer evaluation and language that really that's not transferable. This, is a great surgeon, he's got a great fund of knowledge. Well, what does that mean for a learner? How do I get to mastery? Those are not specific enough objective, details to give me something to work towards. And I think what we're really what I'm hoping for is that we get to some metrics that really define a master surgeon. So if I had to answer the question, are we there yet? Not yet. We're getting there, and there's a lot of technology that's coming up that will actually help me and other researchers in this area. There's sensors that are embedded into clothing materials, microsensors on gloves. And a few of you, I'm sure, have seen all of the hype about Google Glass. It gives us a better ability to capture video. We've had videos in the operating room for a very long time now, but they're in-light cameras. And when we've tried to use it to look at operations, most of the time you're looking at the top of the surgeon's head because their head's in the way. And so now Google Glass or some of the other, we found another sports video glass as well that helps. I actually took these apart to move the camera because they're for bike riders and sports, and so it only faces this way. And when I'm looking down as a surgeon, the field of vision is here when my hands are here. So we took the glasses apart and moved the camera, so now we've got something that'll work for our research. I'm pretty excited. Thank you.